When a patient is actively bleeding, the clinical bedside becomes intensely visceral. Medical professionals narrow their entire field of vision to fighting physics and time right there in the room. Step away from the alarms and walk down to the quiet, sterile hum of the hospital's pathology lab, and that exact same hemorrhage is measured entirely differently. Down here, the crisis is represented by a prolonged clotting time and the absence of a specific molecular cofactor. Vitamin K is often relegated to nutrition labels or vaguely described as the blood-clotting vitamin. In clinical reality, the molecule functions as an active structural switch. To understand this switch, we have to look at its evolutionary origin. Ancient single-celled bacteria used this exact same chemical structure as a highly mobile energy carrier to shuttle electrons around during anaerobic respiration. As complex animals evolved, we adapted this ancient bacterial solar panel. We repurposed it to build the localized biological switches required for high-pressure, closed circulatory systems, networks that can seal a microscopic leak in seconds. Preventing a hemorrhagic collapse requires tracing this molecule's journey from a thermodynamic cellular event all the way to its rigorous clinical laboratory measurement. The active engine of the vitamin K molecule is its chemical head, a 1,4-naphthquinone ring, attached to a variable tail. Deep inside your cells, an enzyme called GGCX harnesses this ring. The reaction releases free energy to weld a double negative charge onto target proteins. This magnetic docking station grabs positively charged calcium ions, anchoring securely to cell membranes to trigger a clot. Yet the total body store of vitamin K in a healthy adult is surprisingly small. We possess a total reserve of merely 50 to 100 micrograms. A single grain of salt weighs more. Because the reserve is so small, the body performs a strict compartmental triage. The liver hogs about 15% of the available supply, strictly to maintain emergency blood clotting. The remaining 85% is left to activate proteins in extrahepatic tissues, like the bone matrix and brain. This limited, triaged reservoir makes the human circulatory and skeletal system entirely dependent on constant resupply to avoid structural failure. To prevent that 50 microgram supply from burning out, the body relies on the enzyme VKORC1. This recycling system relentlessly recharges inactive vitamin K back to its active state thousands of times a day. We feed this recycling loop externally through our diet. Green leafy vegetables provide a continuous supply of vitamin K1, or phyloquinone. Human architecture also features a built-in redundancy system. Down in the colon, anaerobic bacteria constantly manufacture long-chain vitamin K2. They act as a backup generator, providing just enough K2 to sustain the system even if the diet lacks greens. This dependency creates critical vulnerabilities at the beginning of life. Newborn infants are born with a sterile gut. They have no bacterial backup, an immature liver, and poor placental transfer. Without a routine prophylactic injection at birth, they face a severe risk of catastrophic intracranial hemorrhaging. In aging, the risk shifts. As intestinal absorption efficiency drops, the proteins that keep blood vessels flexible are starved of active vitamin K. Without those crucial safety brakes, calcium passively crystallizes in the vessel walls, turning the once flexible arteries rigid and stiff. Human vascular elasticity and structural survival are dictated by the partnership between our diet, our gut microbes, and this recycling loop. If that recycling loop is blocked, the system collapses. The medication Warfarin operates precisely this way. It physically interferes with the VKORC1 enzyme, creating a controlled starvation of the clotting cascade. Commercial rodenticides, known medically as superwarfarins, present a much more extreme scenario. Because they are highly lipophilic, they dissolve deep into human fat tissue. If ingested, they slowly leach out, blocking the recycling enzyme for up to 9 continuous months. Excess states are also dangerous. Oversupplementing synthetic vitamin K3, or menadione, generates oxidative stress that directly ruptures red blood cell membranes. Measuring the status of this system is notoriously difficult. The standard PT-INR clotting test is an insensitive marker for early deficiency. Because the liver acts as a triage center, it prioritizes clotting factors, keeping the PT-INR normal until the system reaches a severe crisis. This flowchart shows how modern pathology bypasses that limitation by looking for uncarboxylated precursors called PIVKA2. When vitamin K runs low, the liver pumps out defective proteins lacking a magnetic docking station. Tracking these duds serves as a sensitive early warning for cellular starvation. Treating normal PT-INR results as evidence of health can mask the silent architectural decay occurring in a patient's bones and arteries. To measure the actual mass of the vitamin in the blood, laboratories rely on liquid chromatography mass spectrometry. This is the gold standard for isolating and quantifying specific vitamin K fragments. The physical chemistry of the molecule introduces a specific pre-analytical vulnerability: ex vivo photodegradation. The active chemical ring acts like an energy antenna. If a drawn blood tube sits on a clinical benchtop, ambient hospital light shatters the molecule's double bonds right inside the glass. When the lab runs that sample, it yields a falsely depressed result, triggering a potential misdiagnosis. Preventing this requires darkroom-style protocols: foil-wrapped tubes, transport on ice, and processing under dimmed amber safety lights. Laboratories also employ automated quality control delta checks. If a patient's INR makes a sudden, massive jump within a 24-hour window, the system automatically halts the result to rule out analytical artifacts or medication errors. The accurate measurement of this master switch is hostage to strict environmental control the second it leaves the body. These analytical vulnerabilities dictate strict clinical stewardship. Medical teams utilize the automated PT-INR assay for routine monitoring, reserving expensive mass spectrometry exclusively for complex diagnostic dilemmas. When monitoring reveals an active bleeding crisis, doctors order therapeutic doses of vitamin K. However, administering that dose via a subcutaneous injection under the skin is a significant clinical error. This cross-section illustrates why. Because the medicine is lipophilic, it gets trapped in the yellow subcutaneous fat layer. It fails to reach the intravenous bloodstream, leaving the patient actively bleeding for up to 48 hours. Vitamin K behaves as an active thermodynamic switch. Total system survival relies entirely on the VKORC1 cellular recycling loop keeping that switch charged. The liver's triage system masks chronic extrahepatic starvation. To measure true architectural decay rather than just clotting times, we must look for precursor proteins through PIVKA-A2 testing. Because the active molecule acts as a light antenna, accurate measurement demands absolute light discipline to avoid critical laboratory misdiagnoses. Longevity research frequently targets human cellular pathways, but the proteins protecting our arteries depend on this ancient imported bacterial tool. Our vascular flexibility is a product of symbiosis, suggesting that the preservation of human architecture relies as much on mastering our partnership with these microbial engineers as it does on re-engineering our own cells.